Good afternoon, everybody. I'm here to welcome you guys also to the Science Influencer Symposium for 2024. My name is Alexandra Ash, and I'm a biology major at Texas A&M International University, which is in South Texas, right on the border to Mexico. This past summer, I had the chance to conduct research at Texas A&M in the Department of Plant Pathology and Microbiology. For a bit of background, sargassum is a brown algae that floats on the ocean surface, and it forms aggregations when it accumulates too much. These large masses of seaweed get washed ashore and block shorelines. And this not only looks terrible, it's very unsightly, but it also smells horrible. The smell is something akin to like rotten egg. So this smell is caused by the seaweed aggregations catching pollutants from the water, and it sort of stays and it leaches those pollutants into the water, and then that's what causes that terrible rotten egg smell. So my main objective of this project was to test whether sargassum could be used to promote the growth of antifungal bacteria. I wanted to test their resistance to FOV4, which is a very prevalent and devastating fungal pathogen that affects cotton fields. And when I say it affects cotton fields, that's an understatement. FOV4 completely decimates cotton fields. And that's what I want to focus on in this project too. It's a way to bridge not only conservation with this sargassum, but also it bridges conservation, microbiology, and agriculture in one multidisciplinary project. First, I amplified the 16S rRNA region of each bacteria strain, and then I performed a gel extraction so I could obtain the DNA concentration through an instrument called a nanodraw. And then we sent the DNA samples off for sequencing and for identification. And once we were able to identify the bacteria's relative taxonomy, we were able to determine the proper media for the growth, which was PDA or potato dextrose agar. This medium is great for both bacteria and fungal growth, which is why it was a good media to use. It wasn't our first choice. The ideal media would have been ISP2, but given the resources that we had, we stuck with PDA media. And so then we got those dried sargassum samples, we grinded them down with a mortar and pestle, and then we added different concentrations of it to the other media ingredients. We autoclaved it, and then we poured it into all these different Petri plates, which is shown up here in this figure. Yeah, that figure there. And as you can see here, the concentrations of the seaweed are 0.01, 0.1, 0.5, and 1%. So after that, I did a co-inoculation. So I pipetted FOV4 into the center of the plate and a bacteria strain on either side. And I used water and regular PDA media for the negative control. So I've just been throwing a ton of methods at you. After all this testing, what are the takeaways? What did we find out from this project? We learned that bacteria strains A and B showed the strongest antifungal activity. So that's these two right here. And you could tell that these are strong antifungal activities because of these inhibition zones right here. There's spaces where the FOV4 isn't growing. And if you'll see over here, this is our strain C that showed no antifungal resistance. The FOV4 was proliferating and growing and took over the whole plate here. So bacteria C had pretty much no antifungal properties. There's a slight declining trend when it comes to seaweed concentration and antifungal bacteria growth. So these findings were the actual complete opposite of what we expected to happen. We wanted to see an upward trend because we wanted the seaweed to positively affect the growth of antifungal bacteria, but we saw a very consistent declining trend. And so that was a very unexpected result, but an interesting one nonetheless. And another unexpected result were the bleaching properties of sargassum, which were present in the 0.5 and 1% seaweed concentrations. You see the fungi growing in the center there. It has a very rosy color in the low concentrations and no concentrations of seaweed. But as we move from left to right, we see that it kind of loses its rosy color here. And so that's why we say it has a sort of bleaching property. We don't know what it is or what part of the sargassum is causing this bleaching property. That could also be a very interesting avenue of research to pursue. And so my personal theory, I would think that it has something to do with the chemical composition of sargassum, since the chemical composition has drastically changed since the 1980s and it's slowly become more toxic over time. So overall, I got very unforeseen results, but that just made me ask more questions, like what would happen in higher seaweed concentrations? What would happen if we try a different media or a different species of seaweed? What would we observe then? So I learned a lot from this project, mainly the importance of patience and troubleshooting, because there was a point in my methods I couldn't get my gel electrophoresis bands to show properly. And that took about a week of troubleshooting because I was, oh my God, what is happening? I'm following these procedures exactly, yet stuff still keeps going wrong. And I think that was the number one lesson I learned. Stuff goes wrong and you have to try a lot of different things to fix it. It was a faulty polymerase and we just ordered new ones. And then things started going smoothly after that. And I want to continue conducting research in the future. And this internship gave me a great framework and a ton of skills to send me on my way for that. So I'm very grateful that I got the chance to do this. And right now, that is it from me.
Thank you
If
so
you guys
much.
have any questions, I will be
Yes.
happy to answer them.